Assalamualaikum and greetings to everyone. We are from group 3. Today we are going to compare two articles which are both about the drought impact on young and mature beach. The title of first article is The Impact of Prolonged Drought on Phloem Anatomy and Phloem Transport in Young Beach. The title of second article is Repeated Summer Drought Delay Sugar Export from the Leaf and Impairs Phloem Transport in Mature Beach. The keywords for the first article are stated there, as you can see in the slide. The objective for the first article is to determine how drought affect carbon transport and phloem transport capacity by combining 13 carbon pulse labeling of 8 years old beech trees. All drought stress trees had significantly smaller sieve tube radii, which reduced sieve tube conductivity and phloem conductance in the tree stem. The hypothesis in this article, trees that are experiencing a drought would absorb 13 carbon more slowly than trees that are receiving adequate water. Next, I will pass to another presenter.
So the second article is based on repeated summer drought delays sugar export from the leaf and impairs phloem transport in mature beech. The hypothesis for this experiment is drought increases the mean residence time of recent photoassimilates leaves of mature beech, and drought reduces the transport velocity in branch phloem. Both parameters were tested for their dependency on pre-dawn water potential and drought stress, those accumulated over the vegetation period. The main objective of this experiment is to determine that repeated summer drought as a delaying effect of sugar transport in phloem transport system of mature beech plant. Next are the keywords. The first is photoassimilate. Photoassimilate is a biological compound that is formed by assimilation through the light-dependent reaction. This photoassimilate is transported throughout the plant's in situ of phloems from the source to the seed. The mean resident time is an average time limit that reason photoassimilate stay in assimilative organs such as the leaves. Additionally, the mean resident times of photoassimilates in leaf increases with accumulative drought season. Then the drought-induced tree mortality are focused on two main interacting abiotic mechanisms such as the carbon starvation and the hydraulic failure. Carbon starvation is defined as depletion of carbohydrate pools as the uptake of carbon decreases under the drought, with plants closing their stomata to minimize water loss. Hydraulic failure is defined as a failure to postulate that tree dies off are largely as a result of dysfunctional water transport that caused by xylem embolism. According to the second article, this research is done by long-term drought stress experiment, which is done in a forest stand with canopy access via crane. This experiment is done in southeastern Germany, and this experiment is to investigate the effect of repeated summer drought of the transport of recently fixed photoassimilates in branches of mature beech European trees, which are the dominant species of temperate European forests.
For materials and method, both articles used appropriate experimental design that can provide data to prove their research. For Article 1, transparent polycarbonate roof was installed above trees. 5 trees from irrigated trenches act as control, and 6 trees from not irrigated trenches were the drought treatment. For Article 2, there were plots with 4 controls and 4 truthful exclusions. Both articles used experimental and control groups to look at change related to an intervention. 13 carbon dioxide labeling method was used in both articles. Decrease in carbon dioxide concentration confirming carbon dioxide uptake. The above ground part of trees in Article 1 were enclosed with transparent cylinder, whereas in Article 2, one branch per beech tree was covered with a transparent plastic bag. Article 2 provided more information on the phloem transport velocity by continuously measuring carbon dioxide efflux and its delta 13 carbon signature. I believe Article 2 did well as the researcher provided adequate information to support the experiment. Both articles calculated pre-dawn water potential to know the plant water status. Article 2 also analyzed the drought stress level by calculating integrated soil water saturation deficit. Next, both articles used the same method to assess the mean residence time of excess 13 carbon in the leaves. Again, Article 2 provided extra method by assessing the leaf non-structural carbohydrates.
To measure mean residence time of excess carbon-13 in phloem extracts, Article 1 used the exudation method. Phloem-soluble compound was extracted from bud strips infused with distilled water, filtered, evaporate, and weighed to calculate the excess carbon-13. Whereas, to measure the mean residence time and time of peak of excess carbon-13 in trunk respiration, in Article 1, trunk respiration and isotope composition was measured using a respiration chamber connected to a trace gas analyzer to measure the concentration of carbon dioxide. Measurement was taken 24 hours before and 96 hours after labeling. While Article 2 does not record any methods for the above measurement. For phloem anatomy, in Article 1, image analysis software was used. 3 stent discs were collected and studied to measure the thickness of the phloem tissue, sleeve tube density, and cross-section area. Whereas, for estimation phloem conductivity and conductance, in Article 1, the hydraulic conductivity of sleeve tube was calculated according to the Hagen-Purcell law, and phloem hydraulic conductance at 3 levels were estimated using equation. Again, Article 2 does not record any method for the above measurement. Lastly, for statistical analysis, data in Article 1 was pooled per tree and 95 confidence interval calculated for control trees. The effect of draft was only taken into consideration if it was outside the confidence interval of the control treatment. While data in Article 2 was analyzed using R. Model was tested for normality and homogeneity of variance. A linear mixed effect model was calculated and data was plotted with the box plot and plot split function.
Alright, for the results part, here it is shown. Okay, in both articles 1 and 2, they mention about the pre-dawn water potential. It means it shows the plant water status. So in article 1, the pre-dawn water potential, it is recorded that the leaf net carbon dioxide assimilation and whole free net carbon dioxide uptake were lower for most of the drought-stressed trees. Meanwhile, in Article 2, it is stated there are no differences in the pre-dawn leaf water potential since a natural drought has reduced the pre-dawn water potential of control trees in late summer. However, in 2016, both pre-dawn leaf water potential and ISWD, or integrated soil water saturation deficit, were significantly different, with control trees showing higher pre-dawn water potentials compared with DE trees. DE is the true for exclusion. Okay, next, as you can see, in Article 1, they didn't mention about the integrated soil water saturation deficit, but in Article 2, they did mention about it. So it is recorded in the results that it was significantly lower for the control trees compared with the DE trees since drought started early during the year and the true for exclusion. Okay, next about the MRT of excess certain carbon in the leaves. MRT stands for mean residence time. So in both articles, they both mention about this, but in two different contexts. So in article 1, they mention about MRT, but they mention it in the 13 carbon enrichment in foliage tissue, flowering step, and trunk respiration. Meanwhile, in article 2, they mention it in another different process, which is the photoassimilation in leaves. Alright, for the next part is focus more on the phloem conductance, but even if both articles have mentioned about phloem conductance, again, Both articles mention it in two different contexts, two different views. So in Article 1, they mention more about the sieve tube conductivity and whole tree phloem conductance. But in Article 2, they focus more on the phloem transport velocity. They didn't really focus on the movement, but rather they focus on the speed of the phloem transportation.
There are similarities between these two articles. Both articles discuss about the effects of drought towards beech trees, while the differences between these two articles are the Article 1 compares trees with control trees, meanwhile Article 2 compares control trees with true fall exclusion trees. Article 1 tests for sieve tube transport conductivity, while Article 2 tests for phloem transport velocity. The weaknesses of these two articles are that Article 1 didn't include much information about the effects of drought as much as Article 2. This is because Article 2 included the information on ISWD as another factor in the research, while for the strength, both articles successfully carried out the research with abundant data and information, and research information provided are easy to understand.
Next, I'm going to talk about the discussion part, where the discussion of Article 1 and Article 2 are quite similar. In Article 1, the carbon transport is slow during drought, where the carbon will export from the leaves to the phloem was reduced. During drought, the phloem figure may decrease, which means it has lost lots of water. Therefore, it reduces the hydrostatic pressure that drives assimilated transport in the phloem, and the phloem transport capacity is also reduced under the prolonged drought. This is due to the elastic walls of the phloem sieve tubes, and water exchange occur between the phloem and the xylem. When the water transfer from the phloem to the xylem during drought, decrease the turgor in sieve tubes and cause a reduction in the hydraulic radius of the sieve tubes. For the Article 2, where during the drought the mean residence times of recently fixed carbohydrate increases in leaves. Sugars are used for osmotic adjustments of the mesophyll cells to maintain functionality and structural integrity during the drought. And during drought also, the phloem transport velocity in the branch was reduced. Where the water in phloem decreases, it will increase the phloem viscosity, which will reduce the phloem transport. And low water availability in the xylem will also reduce the phloem transport velocity that happen due to the flux of water from the neighboring cells into the phloem conduits.
In conclusions, these two articles show the similar circumstances of the drought at it's a young beech tree or for mature beech tree, as generally it will affect the phloem transport to supply the nutrient to the plants. The drought will decrease the velocity of the phloem transport, thus make it slower. In Article 1, it shows that the drought is the reason of the reduction in sieve tube radius, increased inflow, subviscosity, reduced hydraulic conductivity of the phloem sieve tube, and lower phloem conductance at the stem level. And all these effects will decrease the phloem transport capacity. While from Article 2, we all know that the drought will increase the MRT of recent photoassimilates in leaf of the mature beech tree and decrease transport velocity along the phloem pathway that limit trees' capability to respond to drought. Drought will restrict the carbon supply of zinc tissues and defense metabolism that eventually will increase the risk of tree mortality under drought. For example, if roots, which is one of the main zinc organ for carbon, is no longer supplied with sufficient carbon. Therefore, in our opinion, these two articles successfully prove their respective hypotheses. We gain a lot of knowledge from reviewing these two articles, especially about the phloem transport. With this knowledge, more effective action should be taken to decrease the effect of drought on the plant, and it's very important to the future research regarding this field. Thank you.